Everyone, and thank you so much for coming today to our Spatial Technology Symposium. My name is Melissa Olson, and I'm the director of the Genetic Resources Core Facility, or the GRCF. And we're delighted to see such strong interest in this new technology. As many of you know, the single cell and transcriptomics core will be merging into the GRCF. So we are actively in the process of uniting and creating a new arm in the GRCF called Single Cell and Functional Genomics, under the leadership of Dr. Emanuele Palescandolo. Here. He's been very active in organizing the agenda today. So we're excited for this broader effort that we're undergoing. This is stage one. Stage two will be further integration and unification of integrated genomic services across the School of Medicine. Today's program reflects this momentum, and we're bringing together cutting-edge spatial technologies, institutional expertise, and strong partnerships. I encourage you to explore the website of the GRCF to see other services that are offered through the GRCF, future events, opportunities to learn and to train. And I'd also like to take a moment to thank Barb Daly, who's coordinated these events today. And with that, it's my pleasure to turn things over to Dr. Palescandolo. Thank you all for being here today.
Thank you, Melissa. And in fact, as you can see, that partnership is becoming more than that, but it's still a partnership. So my name is Emanuele Palescandolo. As Melissa said, I am currently director of the Single Cell Transcriptomic Core. And as you heard, soon a different name, but basically the same role in terms of delivering single cell spatial activities and GRCF. So this is the agenda for today. As you can see, it's quite short, but it's packed with interesting talks. We try to make it light, but at the same time interesting for you. Thank you for coming, by the way. I'd like to thank, as already Melissa said, Barb and Adrienne to organize this. They did really quickly and very well effort to put this together in a very short time. As much as I'd like to thank Seth Blackshaw, which is the current faculty director of the Single Cell Transcriptomic Core, and Melissa, of course, the director of GRCF. The speakers and you are also a very important part of this. Without you and without the speakers, this wouldn't happen. So thank you for being here again. I'd like also to thank the team of the Single Cell and Transcriptomic Cores, which is listed in absolutely alphabetical order. Each of them has an important place in our core. That's why we are here again, because we deliver to you this. So we think of you as our collaborators. Kind of part of your lab. We like to think about that rather than being, selling kits. That's not what we do. We are here to provide the best expertise possible to maximize the use of the kits. That's why what I say is that the target we are after is the target you're after. As I said already, very quickly, we offer single cell technologies on spatial transcriptomics, also NGS library preparation. And with that, without further ado, I'd like to introduce Brett Toms, which is the first speaker, which is going to give us an overview and a foundation of spatial biology. Thank you, everybody, for being here.
All right. Well, thank you, Emanuele, for that introduction. As Emanuele said, my name is Brad Toms. I'm a science and technology advisor here at 10x Genomics. And the goal of, kind of my talk here in the symposium is to kind of highlight some of the things that these 10x spatial tools can do. Really now with this technology overview, they're really the foundation of spatial biology. So before we kind of begin into that, I always like to kind of start with where we are as a company at 10x and the different applications we have. So we have three main pillars of our technology. All these are available to you through the single cell core. We have our Chromium single cell solution. This is the ability to take that tissue or tumor, dissociate it down to a single cell context, and start to look at it from a multi-omic view. So a very powerful tool for starting to understand those different angles of biology and kind of what is happening within that tissue or tumor. So unfortunately, we don't have time to cover that today. We're really going to focus on our Visium and Xenium spatial products. But if you have questions, please reach out. We can have some discussions at lunch and talk about some of the single cell portfolio. But as I mentioned, I think the main focus today is really going to be on our two spatial platforms. We have our Visium platform. This is the ability now to kind of look at the spatial context of that tumor. So start to understand where genes are expressing. This is going to be a whole transcriptome discovery-based approach at single cell scale resolution. So we'll talk about this today and a couple of the different applications we have within the Visium portfolio. And then lastly, we'll wrap up the day kind of thinking here about our Xenium platform. This is the ability now to look at focus panels, but giving you subcellular resolution. So we can start to go through and find now these dynamics of what's actually happening within that tissue and where those genes are expressing within that spatial context. But for me, in kind of doing these talks and seeing how people are using these tools, it really is a multifaceted approach. And people are going through and using a combination of these tools to go through and start to better understand biology. And we'll talk about a couple papers doing that today. The first one will be talking about using Visium HD in single cell. And then we'll have one in the Xenium section about using Xenium in single cell. And I think this is a great use case here. This is looking at colorectal cancer, so looking at these microsatellite stable tumors. And the thing about these tumors is they are very transcriptionally diverse, right? They're going to be having these heterogeneous transcriptomics profile. And what that means now is because of that heterogeneity, it really now has these varying responses to immune checkpoint blockade therapies. So they needed these tools with high resolution to go through and start to tease apart what that heterogeneity meant for these different treatments. Specifically, they looked at 185 different patients with single cell, a little under 900,000 cells. Some of this was data they generated, some of it was public data sets. So they could start to use the single cell data now as a robust tool for starting to go through and understand cell states, cell programs, immune tumor interactions, and starting to better understand what is happening within these tumors. They're also able to use this data now to go through and perform annotation. So they annotated this data and now could start to layer that back onto the Visium HD. So here, this is just looking at epithelial cells, but now they can start to, again, use this single cell data to come up with these very diverse, very specific annotations that we're seeing down here at the bottom, and then start to map them in space and start to understand what is happening within these epithelial cell populations. So for me, I think it's a really cool, pretty picture here to kind of see what's happening, but they were able to really dive a lot deeper. And I think this is where the paper or the publication really took this turn to starting to understand what is happening within these tumors. And specifically, they wanted to focus on these TAMs, these tumor-associated macrophages. And because it's a whole transcriptomic view, they were able to look at these different subpopulations of TAMs and start to map them in space. And when they did this, they were able to start to see that these are often at the tumor front and kind of in these fibrous strands between tumor cells. But again, kind of seeing where they're at is one view, but because it's a spatial assay, they're now able to start to go through and look at co-localization of these TAMs and start to understand which of these subsets or subtypes often co-localize together and which ones didn't really co-localize very often. And again, lastly, because we have a spatial assay, they're able to start to go through and start to look now at the proximity of these specific subtypes and how close to the tumor they were. And if we look at it closely here, you can see the darker red is the closer to the tumor. You can see specifically in this subpopulation 4, and then the darker the blue, kind of the further away it is from the tumor. So again, looking at all these different aspects, they were able to kind of take this information and start to come up with a phenotype of those tumors and find out which of those tumors really are going to give this anti-tumor immunity, and then which of those tumors or those phenotypes are going to give this tumor promoting properties. And ultimately kind of using this tool as the stratification as to what treatments might best suit those patients. So again, I think it's a powerful kind of view as to how these tools can kind of be brought together to understand the biology in those samples. So with that, I want to kind of jump into what the Visium technology is. And kind of thinking about this, it really goes back now to having this instrument-mediated process. We have the CytAssist here in the lab, in the single-cell core. And the goal here is to use this tool to go through and give us that view of biology in this whole transcriptome discovery-based method. Again, we'll talk about kind of how it works here, but basically we're going to get single cell scale resolution with the ability now to kind of use this barcoded oligo slide at this 2x2 micron barcodes. Some of the kind of other advantages of this assay is that it's flexible, right? You have the ability to work now with fresh frozen samples, FFPE, fixed frozen samples. And if you want to expand in other modalities, you can look at, from that exact same piece of tissue, your H&D image, or you can look at proteins with immunofluorescence. All while giving us now kind of this ability now with free software tools to go through and explore that biology. And we'll be able to do some software demos here later this afternoon. Now, within the Visium platform, we have two different definitions here, kind of these different resolutions. So if you've heard about Visium in the past, kind of the early days was going to be the standard definition. These were 55 micron spots. And when we think about kind of a cell, most cells are going to be a lot smaller than 55 microns, unless you're working with large cells like cardiomyocytes or adipocytes. So this was a great tool for starting to understand those regional patterns, starting to see spatial niches. But it really is not giving that kind of defined resolution. That's where we start to move now into our more recent edition of our HD platform here. This is going to give us that single cell scale resolution. So I always like to show this slide. I think it's a good representation of what that looks like. This is our 55 micron standard definition. We can see here that it's also 100 micron center to center. So there are going to be gaps, right? You're going to have some biology missed. This, again, the way I kind of see it as looking at spatial niches, starting to understand regional patterns. But for those studies that need to dive a little bit deeper, that's where the H.D. comes into play. And these are consecutive sections. And now you can see what that jump in resolution is allowing, right? There's no gaps here. We're able to capture that full biology. We're able to now get much more kind of defined spatial clustering because we're not missing anything, because we're able to get that higher resolution. So the majority of today's discussion really is going to be focused on HD. Again, there are going to be applications where standard definition makes sense. So if you have those, please reach out. We can have those conversations. But I wanted to kind of take a step back and think about not just what we can do with the technology, but how it works. And with this, we're able to work now with these uniquely barcoded slides. So within the CytAssist slide, we have two capture areas. They're 6.5 by 6.5 millimeters. And within that 6.5 by 6.5 millimeter capture area, we have about 11 million 2 micron spots. Now, for sake of data analysis, we typically bin them to 8 microns, but you have the raw data that you can go back and start to dive deeper should your scientific question really kind of need to be down to that 2 micron level. Now, again, this is going to be a continuous lawn of oligos. So each of those 2 micron spots is going to be now a lawn of oligos that is going to have very distinct kind of a pattern here. And this is what allows us to get down to that single cell scale resolution in the spatial aspect. In black, it's a PCR handle. In orange, it's a UMI. This is a unique molecular identifier. It allows us to count unique transcripts without counting PCR duplicates. In green there, we have a spatial barcode. This is going to be really what's allowing us to track things back to that spatial level. This is a unique DNA sequence for each of those 11 million 2 micron features. This is what I like to think of as almost like a GPS address. Because on the end there in blue, we have this poly-DT capture sequence. That's going to capture our probe information or our native poly-A tail on our mRNA transcripts, depending on which workflow we're working with. And then we use molecular biology to tag that UMI and spatial barcode onto the transcript. Informatically, then, we can go back now and map that back to that exact 2 microns feature that read came from. So within the Visium HD workflow, again, we want to give you options, right? We know there's no one-size-fits-all approach to biology. We have different approaches here. We have our whole transcriptome panel-based approach. Because of this is going to be able to work with more degraded samples, such as FFPE. Here, instead of capturing by a poly-A tail, where degradation could really kind of make that sensitivity really start to drop here with those fragmented samples, we're going to use this whole transcriptome probe set where we don't need large real estate to go through and hybridize those probes. We can hybridize those probes now and start to capture that transcriptomic information for those highly degraded samples. But because it is probe-based, we have now these comprehensive panels. They're going to be for human and mouse. But we know there's a lot of biology that needs to be done outside human and mouse. This is where I came up with our Visium HD 3' approach, where we're going to go through and capture by that native poly-A tail, really now making this species agnostic and giving the potential for other aspects. If we want to pair this with long-read sequencing, looking at things such as isoforms, SNPs, potentially even TCR, VCRs in the context of the tissue. Now we really want to start by thinking about that probe-based approach here. This is our Visium HD whole transcriptome probe panel. This is going to be the ability, again, to work across a wide range of samples, FFPE, fresh frozen, and fixed frozen, all at this single cell scale resolution. And it's going to be compatible, again, with, from the exact same piece of tissue, the ability to do H&D or immunofluorescence staining. And kind of what we look at here is kind of the overall view of what this particular assay works with. The high level view is we're basically going to take that tissue, perform a sectioning, right? Section that tissue onto a pretty standard glass slide. It's at this point we get to stain and image it. So I think this is really critical, especially for a lot of biology, right? We need the ability to preview that sample. We can put it on a slide, we can H&D stain it, we can make sure that it has those features you're looking for. And if it doesn't, it's just on a glass slide. We can set it aside, move forward to that next section, make sure that we do have that tumor profile or that right plane or the necrosis you're looking for. Once we've previewed it, then we can decide now we want to start moving forward into the rest of the workflow. This is where we'd make our RNAs accessible. We would hybridize this whole transcriptome probe set, and then we can move forward into the assay. Here at this point, again, this is where it goes into the CytAssist, where the CytAssist is going to essentially take those probes from your tissue on your glass slide, transfer them to that Visium capture oligo slide. From here, pretty simple molecular biology, making an Illumina-ready library into sequencing and then data visualization. And when we think about the whole transcriptome probe set, I'd really like to highlight this and kind of in what it looks like here. With this, again, it's going to be about 18,000 genes, so a little over 18,000 in human and a little over 19,000 genes in mouse. But in total, in order to kind of help increase the sensitivity and specificity, we're looking at multiple probes per gene. So it's a little over 54,000 total probes. So we're just showing it once here, but again, remember, this is starting to happen at scale with 54,000 probes. What we have in this probe set, we have a left-hand probe in purple, right-hand probe in gold. They're going to be very specific for those mRNA transcripts where they will go through and hybridize. And we need them to hybridize right next to each other so we can ligate it together to create a single-stranded probe pair product. This is important for that specificity. This is not one random probe that could hybridize non-specifically. We need these multiple events to happen, right? We need the left-hand probe to hybridize, the right-hand probe to hybridize, and they need to be next to each other so we can ligate it together. Otherwise, we're not going to get a signal. So this is done intentionally to increase that specificity. The other thing you'll notice within that right-hand probe on gold, we have a synthetic poly A tail. This is how we're going to capture this information on the Visium slide. So when I think about this panel, again, human and mouse, comprehensive whole transcriptome, but we know there are genes that are missed. Sometimes people want to add in targets for looking at viral targets or exogenous sequences, such as those viral targets or reporter genes. So if that, we do publish the genes as well as the sequences of these probes online. If you find you have a gene missing or you want to go through and add those additional exogenous sequences, we have a tech note for going through and kind of creating custom probes and how we can spike those in to really start to go through and look at specific genes that you might be interested in combination with that whole transcriptome probe set. So as I mentioned, again, what we'll get to on this tissue, this is, again, with the tissue on your standard glass slide, we have introduced our probe sets. It's at this point, we take those input tissues, they go on the top of the CytAssist here. We take our Visium HD slide, it goes down below. And when we close that lid, it brings your tissue in a very close proximity to oligos printed onto that slide. This is where we permeabilize the tissue. It's going to release those probes. Those probes then will use that synthetic poly A tail to bind to that poly DT capture sequence on those oligos. Looks a little bit like this. So you can imagine we have our slide over on this side. We have that poly DT capture sequence. That synthetic poly A tail will hybridize to that poly DT capture sequence, where we can then just use a pretty simple molecular biology, do a DNA probe extension, and start to tag your gene expression information with that spatial barcode in UMI. And it looks a little bit like this. So here's a couple examples. And the thing I always like to point out about this is that this is not, a microscope image. This is going through and looking at this unbiased spatial clustering. So starting to look at those spots on that slide and starting to find now which spots are going to be most transcriptionally similar, and it's going to color code them. And what we can see now, looking at some of the germinal centers in the tonsil, we can start to see with the mouse embryo, you can start to see the keratinocytes here, starting to see that whisker development, the heart development. So you can see even in the brain and in some of the palate. We can start to go through in a transcriptomic view, start to understand kind of the morphology and kind of the biology of that tissue. Now, with our FFPE workflow, we validated a wide variety of samples, especially even challenging samples such as TMAs. Imagine we can layer in now biopsy cores and start to bring down that cost per sample by putting multiple samples within that capture area. We can start to work with difficult samples that maybe have high RNase content like pancreas, so we can go through and fix that and kind of deactivate some of those RNases. But again, working across a wide range of human and mouse samples. And we have some of these data sets online. As far as fresh frozen and fixed frozen tissue, we've also gone through and validated, again, this workflow using the probe-based approach in combination with other sample types. So we give you, again, that flexibility. You have the ability, if you're collecting these samples, to choose any of these three methods, depending on what works best for your sample type. Or if you already have samples collected, then you have them archived and you don't have a lot of flexibility kind of as to what workflow you would need to work with. But we have options here and the ability now to kind of move forward in that transcriptomic view. So with that, I did want to kind of do a small pivot now to thinking still again about Visium HD, but thinking about the 3' approach here. So this is going to be capturing by that native poly A tail. And the goals here are still going to be the same. This is still now this whole transcriptome discovery. But now, because we need to capture by the poly-A tail, we need those mRNA molecules intact. The samples need to be fresh frozen. Still using that same slide, so still going to have this 2 by 2 micron barcoded oligos of about 11 million 2 micron features, and it's still now enabled by the site assist. With this, and I know there was some early on adoption with the Visium fresh frozen workflow here at Hopkins. We had a workflow before using our standard technology. And I think while this did provide some great insights, there were often challenges with the Visium fresh frozen approach. Specifically kind of thinking about these challenges down here. The first one, 55 micron spots. Again, good for spatial niches, regional patterns, not giving you that resolution that a lot of researchers needed. The other one was direct placement. We had to take that tissue and directly place it onto that capture area. So you were committed. You lost that ability to kind of preview that sample. So, and then the last one would be tissue optimization. We had to go through and permeabilize completely through the tissue, meaning that each tissue you worked with, even within the same tissue in different disease states, we had to go through and find that optimal permeabilization time. Well, now we've moved to HD3', we've kind of overcome now a lot of those limitations, right? This is now single cell scale resolution. This is now site assist enabled. So we're going to use that instrument as this mediated process for transferring those mRNA molecules to the slides, making it now, again, something where we can preview those samples ahead of time. We can start to get now a precise transcript transfer and localization. And then lastly, that single protocol, no more tissue optimization. So it really makes this workflow a lot easier to get into. High-level view still looks very similar to what we talked about with the probe-based approach, where we're going to section our tissues onto that slide, then move forward into the site assist workflow, where actually we will do our staining and imaging on that sample, make sure it's something we want to proceed with. And then at that point, able to kind of move forward into the site assist workflow, where we can now, again, transfer those poly-A transcripts to the slides. And now, because it is a poly-A-based method, we're going to use different approaches, but still very simple molecular biology, such as reverse transcription, to tag those transcripts with your spatial barcode in UMI. And then again, using our free analysis tools, start to go through and map them back in space. As I mentioned, because this is fresh frozen, or because this is a poly-A-based capture, we really want to ensure that we are capturing now high quality mRNA molecules. So we are going to work with fresh frozen samples and fresh frozen samples only. And this is compatible with H&D. So if you have fresh frozen samples that you want to work with immunofluorescence, we need to move to the probe-based approach. But again, I think this opens up this species agnostic view where you can see we've done testing here on, of course, a wide range of human and mouse samples. But I think what really is opening up now the new door is kind of these other species here, a lot of non-human primate research, rats. I believe we have some xenograph, pigs, chickens, zebrafish. So this is kind of now enabling now the single cell whole transcriptome discovery method for those species outside of human and mouse. And with that, you can kind of get a quick view here. This is a fresh frozen rhesus macaque kidney, where we can start to go through and see what some of that spatial distribution looks like, looking at different canonical markers. I think the immune cells, we're looking at B cells, the distal tube cell markers, and of course now these proximal cell tube markers. So start to kind of see, and unfortunately the images aren't coming through real well here, but we can overlay this with H&D and kind of match up where those immune cells are at within that tissue. So, kind of thinking about those two platforms, I know 10X is big about innovation and kind of driving the biology and these discoveries forward, but it isn't just on the wet lab side. We really want to enable now kind of this end-to-end solution. And with that, we also are kind of innovating on the analysis side. So these slides are not showing up very well. As we can imagine here, the Loop browser here, this is our free analysis tool. So we have primary analysis with our Space Ranger, and then with our Loop browser, we have secondary analysis. This is a free tool. We'll do a demo later this afternoon, but the ability to start to kind of understand that spatial clustering. You can also pair this up with your H&D or immunofluorescence image, where we can start to view where those transcripts are at within the tissue. We can perform local differential gene expressions, violin plots, do manual annotations. We can import annotations. It really gives kind of this nice, what I call, again, secondary analysis view into that data. Start to understand the biology and what's happening within your tissue. But as I'm sure you've heard me say a couple times today, this is single cell scale resolution. And the reason why we say that is, again, we have these 11 million 2-micron features. Your tissue is not going to line up over a perfect grid of 11 million 2-micron features. So for sake of analysis, we find most cells about 8 microns. So we do 8-micron binning. This, again, your tissue is not going to line up perfectly over that. Some bins, like this purple one here, that might be single cell resolution, right? We only have a part of that cell on here. We're not sharing multiple cells within that spot. Again, probably single cell resolution down to this guy. But you're going to find now where some of these cells start to intersect, we're going to find now these bins that have multiple cells in them. So again, this is where we call the single cell scale resolution. But a lot of researchers started to see the value in kind of having that high quality H&E image and using that to identify where the nuclei are at and then going through and rebinning. So this now gives us a little bit more confidence in that single nuclei transcriptomic profile. But as you can imagine, now we started to lose a lot of those transcripts that are out in the cytoplasm. This is now with our most recent kind of innovation on the analysis side here of going through and performing cell segmentation. The ability to use that high quality H&E image, identify where the nuclei are at, and then do an expansion off of that. Now we'll talk about segmentation stains with Xenium. Those are going to be true morphology stains. They're going to be an imaging-based output. That is going to be much more definitive than this, but this is giving us now an improved resolution. But because you can imagine here, I can't see it from this side here, but there's going to be some of these bins here that will still have a little overlap. This, again, is not going to be perfect segmentation, but I think has really started to go through and improve the resolution. And I've heard some pretty powerful statements from customers on how this is improving the data. With this is a free software tool, as long as you're running the most recent version of our Space Ranger 4.0, as well as our Loop Browser 9.0. Both free softwares, so you can really jump into doing this and performing these analysis, run seamlessly in the background, where it'll do your 8-micron binning, as well as this cell segmentation view. And in kind of thinking about what this looks like, we can see a couple different sample types here, a pyre patch, as well as intestinal villi with the H&E image. We can see what that looks like with standard 8-micron binning. Getting a nice, decent view of biology, but you can kind of see that, kind of in these areas, it's still a little ambiguous, right? We can't get that true definition of what's happening. When we perform these segmentation-based clustering algorithms, we can now get to see kind of more now what's happening beneath the surface, right? What is happening now definitively kind of in these different regions, and we get a much higher resolution image. And I think the power of this is you really don't need to do much different, right? This is a free analysis tool. It's going to run in the background. The only thing you need is a high quality H&E image, which you should have anyways for moving forward into this workflow. Other than that, these two are optional. And if you want to move forward with these optional aspects here as well, if you're working with a non-human or non-mouse sample, we typically trained the software tool on human and mouse. If you have something that kind of goes outside of that, you can create your own segmentation mask and import that. And again, with the bottom one here, we actually use that H&E image to identify the nuclei and then do an expansion from that. And you can kind of pick and choose and customize that expansion that you're looking for as well. But again, those are both optional. The only thing you really need is a high quality image. And with the outputs, you're going to get that now in the standard analysis of a loop file, your JSON files, as well as web summaries. And now what we start to see is a little bit more, instead of kind of these perfect grids, we're just going to start to see this true representation of biology, where you're going to have some cells that are smaller, some cells that are larger. So again, this is kind of that view of the loop browser. But I think the real reason I find value in this is kind of really thinking about a view like this, right? If we're looking at CD4 T cells. So with T cells, they're typically pretty small cells. They're typically not a lot of high RNA content. So if you find them kind of sharing an 8-micron bin, and it's sharing that with a cell that might be a tumor cell that's going to have a much higher transcriptomic profile, they can often get drowned out, right? You can kind of see here the green and CD4 T cells only finding a few spots here. It's probably because they're overlapping some of these bins with other cell types that have much higher RNA content and kind of masking them. They just kind of look like background. When we apply these algorithms, you can see now we start to get a much different picture of where those CD4 T cells are at, because we can identify the nuclei and then start to capture just those instead of sharing those spots with other cells. So again, I think a pretty great tool to kind of go through, especially in these early analyses, start to go through and look at 8-micron binning, look at these different cell segmentations and see what answers your scientific questions the best. So with that, I kind of want to do a pivot now to think about our other spatial platform. And this is going to be our Xenium platform. This is the ability now with a morphology-based readout. So again, moving away from that approach with Visium, where we use now the sequencing-based approach, we're going to get now this microscope image where we can start to see where those transcripts are at in situ, or kind of in the subcellular resolution. With this, we have a lot of key differentiators within this platform here, specifically with our probe design, where we can get this very highly sensitive, highly specific. We can look at SNPs at subcellular resolution, look at isoforms at subcellular resolution. And it's all because of this specific probe design, which we'll touch base on. The other one is again, kind of now this flexibility. We have the ability to go through and create custom panels. We can look at non-human, non-mouse species. We can look at smaller panels of genes up to 480 genes, or we can look at panels up to 5,000. So you give now this flexibility that you can kind of fine-tune your experiments or fine-tune which assay you run based off of what those scientific questions are. And of course, I think things that are really important for the core is the fact that we can put a lot of samples on these slides and has a quick turnaround time. So we can start to move through some of these larger projects. But what does the data look like? Yeah, I think this is kind of that powerful view, right? We're getting this microscopy view. We're getting now the ability to see those transcripts in situ or in the cell. You can see down here in this example, we have a macrophage here in purple next to 2 T cells. If you look closely, and it might just be hard to see here, you can actually see kind of through this opacity, we can see that DAPI-based stain where we can see the nuclei. We're getting sub 30 nanometer XY lateral resolution, where we can now start to identify those transcripts, which ones are in the cytoplasm versus which ones are in the nuclei, and start to get this subcellular view. So a great publication on this, again, kind of looking at this multi-omic view, is kind of understanding these immune stromal niches here within a dermatomyosistis sample here. So I apologize. I just read this paper yesterday, but I thought it seemed like a perfect fit. So we'll kind of stumble through this one. But kind of within this study here, they ran, again, 4 different samples, these dermatomyosistis samples. CLE, this is going to be your cutaneous lupus samples, as well as healthy and non-lesional. They ran this multifaceted approach we talked about, where we're going to run 44 samples within a Xenium workflow. And then within our single cell flex workflow, ran 17 samples. And they use this data to perform these data integrations. You can see down here on the bottom to look at mechanism of action, to start to perform these integrated data sets, annotations, understanding now these spatial niches. So high level view, if you look at this DM sample versus now these cutaneous lupus samples, if you look at them under a microscope, they're quite similar. But now when we start to look at them kind of at this molecular level, they're going to be kind of these different patterns here. You can see healthy versus the DM sample, and then this cutaneous lupus sample. And what we can see mainly is going to be on this immune and vascular regions, right? We can see a little bit of that up here in the DM, maybe even kind of speckled through the rest of the tissue, but really now a different profile in that lupus sample. This is where I think the value of that technology lies, right? It's things you can't really kind of see from that microscope level, but now we can start to go through at the molecular level and start to understand what's happening. And ultimately, when they really started diving deep into this, they found that the DM lesions, these really had now these areas that were infiltrated by neutrophils, so a lot more of that immune infiltration, which was kind of controlling some of those tumor samples. But those neutrophils, they were absent in the lupus sample. And if we kind of look at over here, you can again kind of see your neutrophils here in the DM, but kind of absent in the CLE. Where if we look down the CL, actually the Langerhans cells as well, I think was another one that they were standing out here. There we go. These guys down here. When we start looking at plasma cells, B cells, these were now those cell types that were more prevalent in the CLE and not in that dermatomyosistis. And then again, diving deeper, again, we have now, I think they ran the 5K panel on this. So we have 5,000 genes to look at, starting to look now kind of at this non-cancer versus cancer, and starting to see these immune niches where I think kind of now using those immune cells to start to control why they were non-cancer versus allowing that cancer to kind of move rapidly through the tissue. Ultimately finding therapeutic targets, looking at interferon beta, as well as a couple other markers now that could be used to kind of go through and essentially, since these are skin biopsies, is a tool to molecularly look at these lesions, look at these different sample types, and be able to use it as a stratification for these patients as to which therapies they might be better suited for. So ultimately, I know there's a lot on this slide. I think the value props here in bold, single cell, really starting to go through and identify those cells, starting to understand those different transcriptional profiles at a whole transcriptomic view. And then kind of moving to the Xenium side here, where we're looking at a focus panel. 5,000 genes is still a lot, but it's not that full transcriptome, right? That's where we're using a lot of the single cell data. But now going through and starting to map those where they're at in space, starting to look at those cell-to-cell interactions, right? You know, that tumor cell next to an immune cell is going to have a different transcriptomic profile than a tumor cell that might be in kind of now a very specific tumor niche here, surrounded by other tumor cells. And then, of course, now linking that immune response and that inflammation to that tissue damage. So hopefully that was a high-level view of what you can do with these tools, but I wanted to kind of take a step back for some of the new users as to kind of how things work here. High-level view, again, is a pretty quick workflow up front, pretty standard sectioning, histology work, standard molecular biology, but essentially working now with a microtome or cryostat to section your tissue onto the Xenium slide. We then make those RNAs accessible, where we can go through and introduce our probe sets, and then we can perform that molecular biology. So in a little more detail here, again, we're working now with a two-probed approach. We have a green probe here on our left, and on the right, we have now this red probe. So these, again, are going to be very specific for that transcript, where we will hybridize them with the goal of ligating them together again, right? We can use this to increase that specificity. The main difference here and between the Visium ones is we have this handle or padlock design. And within that, we build this gene-specific barcode. So now once we've ligated together, it's a circular product. So we can use a rolling circle amplification, and we can start to boost up now that signal. We're not as concerned now as much with where that probe hybridizes, but we're concerned now with using that RCA to kind of create these little kind of DNA balls here, where we're going to go through and have multiple copies of this gene-specific barcode. So this is the upfront workflow. Once we have this ready, we can take 2 slides, load them into the Xenium instrument, hit start, and it's going to run for a few days, depending on if you're working on our V1 panels or our 5K panels. But what's happening inside the instrument is it's going to essentially walk down this gene-specific barcode with fluorescent oligos. We'll bind a fluorescent oligo. We're going to capture that optical signature. We then strip off that fluorescent oligo, add a new one, capture the optical signature, strip it off, add a new one. We're going to do this iterative process somewhere between 15 and 36 times, depending on the assay. And what we can do now is we can come up with this optical barcode. We're going to go through and look at each cycle, figure out what fluorescence lit up, and come up now with a pattern. Now we have 4 fluorescences, and we have 1 dark signal, so no signal. So we only have 5 options. So you can imagine looking at 1 cycle is not going to be informative. Looking at 2 is probably not informative. 3 is probably not either. But as we start to build that over 15 or 36 cycles, we can create a unique pattern and kind of map it back to the known expected profile of what we're looking for. I've heard somebody recently kind of compare this almost like Morse code, right? We can go through and look at the combination of those different signals to kind of come up with what that sentence is. Here, we're going to look at those fluorescences in each cycle, really come up now with this unique fluorescent barcode. Again, some of the advantages here is this high sensitivity, high specificity. We'll use that probe-based approach now with having to ligate them together. If there is a mismatch at that ligation point, we're not going to get a signal. We can't ligate it if we can't perform that rolling circle amplification. So this gives us a high level of specificity. And because of that specificity, we have the ability to look at isoforms. We have the ability to look at SNPs. The probes are small, so we have the ability to work with highly degraded samples. And I think this bottom bullet point is one of the more important ones here that often gets missed. We have the ability to fine-tune custom panels for your specific biology. So when you create a custom panel, we ask for single-cell RNA data, and that allows us to understand in your sample which genes are high expressors, which ones are low expressors. This gives us the ability now to essentially layer the number of probes we want per transcript. Remember, we're not interested with where they hybridize. We're interested in that gene-specific barcode. So in this example, we have a transcript. We put 4 probes on there. So now we have the ability to kind of boost that signal. So we have the ability to tailor anywhere from 1 to 8 probes, meaning we can take a high expressor and use 1 probe, so kind of dampen that signal down, or we can take a low expressor and use 8 probes, and we can kind of boost that signal. So we get a very large dynamic range within these samples. Another great differentiator is the ability to do custom content. We have off-the-shelf panels. You can see here about 380 plex for human and mouse. But we know that there's no panel that really fits everybody's needs. We can't make everybody happy. So we give you the ability to add in 100 custom targets that might be more specific to your scientific question. Now, 480 genes is not enough. We have our 5K panel for human and mouse, again, with the ability to add in 100 custom targets. So you can kind of customize that. And again, if those still don't fit your needs, if you're working outside of human and mouse, if you want to look at some advanced customization, we have the ability to do full custom panels across a variety of species. I have researchers that have done mosquito panels, non-human primates, dogs. We can go through really, as long as we have a well-annotated single-cell reference, can create custom panels up to 480 genes. And with this, because of, again, that unique probe design, we have the ability to do this advanced customization as well. And I think this is, again, one of those things that sets us apart. The ability to look at isoforms, right? That's subcellular resolution. The ability to look at SNPs, again, the single base pair variants and how we can see that at subcellular resolution is quite powerful. And then, of course, looking at exogenous sequences, viral targets, reporter genes, xenografts, where we can kind of now have the ability to distinguish host versus graft. And then if you know TCRs, BCRs of interest, the ability to go through and see that adaptive immune response at subcellular resolution. So we have several off-the-shelf panels. This is just one of them. This is our human immuno-oncology panel. And the goal here, again, is to kind of go across these wide ranges here of what we're looking for, right? Cancer biology. So start to understand these tumorigenesis, these other aspects of cancer. Start to understand the immune cells, right? As we start to see within that tumor microenvironment what that immune response is. Cell stating, cell function, right? We need to kind of understand where those cells are at kind of within their cycle. And of course, still we need to see the kind of the structural aspects of that tissue. And again, with this, you can add in 100 custom genes as well. If you really now have specific questions of your own, or you want to build this for specific TCRs and see where they're at in space. The other panel I just wanted to highlight real quick, again, is our 5,000 plex panel. This now starts to blend that kind of view of focused versus discovery right now, as we can start to expand the number of genes we can go through and address, giving us now this highly kind of scalable solution to kind of look at more genes, more targets. But I think kind of having a marketing slide up there saying we can do all this advanced customization is one thing. I think it's great to see it from researchers such as yourself. You can see here is exogenous sequences, looking at 2 viral targets where we created probes for, I think it's HPV, HPV, and then human infected COVID lungs. Organoids can often be difficult to work with. So we can see down here on the bottom, looking at now kidney organoids. Here in the middle, I have a better example of this, but this is Simon Gregory down at Duke, where he was looking at glioblastoma and looking at isoforms at subcellular resolution. And of course, down here in the bottom, I believe this is a publication out of Asia looking at colorectal cancer and looking at these single nucleotide variants, starting to understand kind of that SNP approach here and kind of that SNP view of what's happening in colorectal cancer. And then for those researchers, again, outside human and mouse, you can see tilapia brain, looking at corn as well. And, kind of briefly within, how this is working with isoforms, again, because of this 2-probe design, you can see colors might not be showing real well, but this is a yellow exon, a green exon, and a red exon. Because we have this 2-handled approach, we can sit down 1 probe on one side of the splice junction and 1 side on the other splice junction and create this now for these different isoforms. And again, because if it's not sitting down to the point where we can ligate it together, we're not going to get a signal. So we're only capturing that information for that 201 isoform there in red, and then for the 202 isoform in green. And again, I think while seeing it on a 10x marketing slide's interesting, I think the real power comes when you start to see it from researchers. This is Duke research here, where they created, I believe this was glioblastoma, looking at this EGFR splice variant. They created a probe spanning exon 1, exon 2, exon 7, exon 8, and then exon 1, exon 8. And starting to see these isoforms at subcellular resolution, I know is really critical in such diseases as glioblastoma. Another differentiator of the technology is we have a very large imageable area, this 10.5 by 22.5 millimeter imageable area, where you can layer in samples really in any format that you want to. We're going to be able to capture subcellular resolution for everything within that imageable area. Meaning if you're working with a large tissue, you can fit that into this imageable area. If you're working with something such as a mouse brain, I have researchers that have put 4 mouse brains. So now you can kind of do an N of 2 across 2 different conditions and almost have like a mini pilot experiment all on one slide. And then for those researchers that work with biopsies that have TMAs, you can go through now and start to kind of create these TMAs to really maximize that and drive down that cost per sample. I've a researcher at NIH that I think did a TMA of, it was 4 by 10, so it was somewhere 4 by 12 maybe, so somewhere in that 40 to 50 TMAs, and they could go through and look at different drug treatments. They could put their controls, they could build in different tissue types, and really do a scientific experiment over 1, maybe even 2 slides. Another thing that's I think really stands out about this technology is the ability to do cell segmentation. We talked about it on the Visium, where we can kind of identify the nuclei and expand. It's powerful to a point, but it's not perfect. Well, with Xenium, we have this ability to use this now in a morphology-based readout. And I think this is really critical because if we think about subcellular resolution, that's really only good if we can tell where one cell ends and the next one begins. And with this, we have a multimodal segmentation cocktail. I'll show this in the afternoon, what this looks like in our software. But basically, it's one tube with all these different stains where we can go through, find the boundary stains. That's going to be the most definitive, right? If we can stain where that boundaries are at. If that doesn't work, we go to the 18S, right? This is going to fill up kind of that cytoplasm now so we can start to infer where that membrane is, where that nuclei is. We have interior proteins in the mix. It's not used in the segmentation algorithms at the moment, but it is in the cocktail. But so that last approach then would be if it's not being called with any of these other methods, does a DAPI-based stain, identify the nuclei, and then you can set that expansion, no expansion, all the way up to 15 microns. I think the default is at 5 microns. So a little bit about what that looks like. You can see we have our tissue here. Boundary stain, this is going to be the most definitive, right? If we can stain where that boundary is at. This gives us the ability to look at cells within one nuclei, looks at multinucleated cells, and even cells that don't have a nuclei. If we can't call that, the next step down in that kind of hierarchical approach is going to be using that 18S. Here, again, we can kind of start to fill in where those transcripts are at in the cytoplasm. Now we can infer where that boundary is at. With this, we need to have a nuclei stain there in order to make sure we can call that appropriately. And then if that doesn't work, we basically identify the nuclei and then do an expansion out. And as with everything here, we've gone through and tested across a wide range of human and mouse samples. So we have kind of this cocktail that works across a lot of different sample types. So I'm not going to say it's perfect for every sample type. So definitely reach out and have these conversations with the core, have these conversations with 10x to make sure these markers will work for your sample type. But across a wide range of tissues, this is really now giving us the ability to do that segmentation. If it doesn't work, the software is open-ended. You can use segmentation methods that you currently use for your studies and then use that to segment and then bring it back into our tools as well. So there's a lot of flexibility. People use other transcript-based methods as well, like Bayes-or ProSeg, where you can look at the transcripts that are present and then do segmentation based off of those canonical markers. So lots of different options. And then thinking about this technology, it's non-destructive. So we can go through and take that slide out of the Xenium and perform applications post-Xenium run on the exact same piece of tissue. And again, this is where I think it starts to come into this multi-omic approach that we see with these 10x tools. This is an example of a post-Xenium run IF stain. So we do have a tech note on this. The tech note goes through and kind of walks you through on how you can run these orthogonal technologies post-Xenium run. With that, we validated H&E staining, immunofluorescence staining, as well as the Visium site assist. Now, with immunofluorescence, we validated this as a single staining and imaging event, so typically 4 to 6 markers. But we know researchers really want to go beyond that. And this is where I think kind of the research community really is taking these tools and started to use their own kind of ingenuity here and start to expand on that. Jack's lab out of Connecticut came up with Protocols.io, so this is available online, of how to take that slide out of the Xenium, and they ran a 40-plex protein panel on the Coit phenocycler. We can kind of do this image registration, bring these images together, and start to see now highly plexed, multiplexed immunofluorescence data on top of subcellular transcriptomic information. It can provide a really powerful tool of biology. Now, with that, kind of thinking about, again, these multimodal aspects here, we also recently came out with a Xenium protein panel. And why is protein important? Well, I think, if we look at biology, gene expression and protein expression does match up sometimes, but there's going to be certain markers where they don't match up. And this is where I think having these robust multi-omic views helps better understand biology, right? If we can look at gene expression and protein expression from that same tissue. Now, other aspects such as biomarker validation, therapeutic targets, these are often also kind of associated with the protein information. But having that RNA information can really add some benefits as well. So having this multi kind of faceted approach, I think, can provide these insights. What's nice here, it adds a little bit of time to sample prep where we need to stain with those antibody panels. It's going to be part of the Xenium readout, right? We're going to put those slides in, hit start. It's going to get your transcriptomic information. And it's going to do additional cycles for capturing protein. And then this is compatible with the first protein panel we have out is a human immuno-oncology panel, basically looking at our human V1 assays with human FFPE samples. So a lot of those off-the-shelf panels, that immune-oncology panel I showed, custom panels, you have this flexibility, but it needs to be one of our V1 panels. And within the proteins, we have 27 different proteins, and we have them in modules. So you can kind of pick and choose and stack these modules with the proteins that make the most sense. Using anywhere from one to that, I just want to say, all right, there you go. Thank you.
All right, thank you, Brad. And there's any quick question, because Brad's going to be here also later for a Q&A with the team. But if you have a burning question about any of those, please fire it up now.
Hi everyone, I'm Angela and I'm a graduate student at Dr. John Ling's labs. And today I'm going to talk a little bit about how we're exploring the upstream mechanisms of TDP43 dysregulation with the 10x transcriptomics technique. So a little bit of background, our lab is interested in a protein called TDP43, and its main function is to repress the cryptic exons, which are these regions in red. They are actually normally introns, and they would get spliced in when TDP43 loses its function. So for example, in this example, the ground truth of the percent spliced in is 50%. And there's a mechanism in the normal cell that's called nonsense-mediated decay that could detect those basically wrong transcripts and degrade them. So when NMD is at its 100% efficiency, you will not observe any of those cryptic exons being expressed. So the observed percent spliced in would be 0. And in contrast, when we inhibit NMD and it's at lower efficiency, much more transcripts with the cryptic exon will be expressed, and you will see a higher percent spliced in. And it's previously established that there are a lot of cryptic exons, and different papers have a different ratio of the in-frame cryptic exons, meaning that the insertion is in multiples of 3, so it would be in frame of the normal transcripts. And these are some of the example, so there's a lot of the cryptic exons. And so Irika, a former graduate student in our lab, and Ing, a graduate student in Dr. Shuing Sang's lab, have collaborated to basically knock down TDP43 and also inhibit NMD in I3 neurons, and those are iPSC-derived human cortical neurons. And they want to basically, by inhibiting NMD, reveal the cryptic exons that are normally hard to detect. And these are, so they used shRNA against these 3 genes that encode proteins that are important for NMD inhibition, and found that they found around 393 cryptic exons, and they have a lot of different categories of exons, cryptic exons, and around 5% of them are in frame. So these are just some example expressions. So in the control and when there's no TDP knockdown, and when there is TDP knockdown and without the NMD inhibition, the transcripts look similar. But when you start inhibiting the NMD, the cryptic exon, which are labeled by the blue arrows, start to be expressed. So for our so how we used Visium HD was that we used human FFPE tissues, and we first stained them with IF staining with the TDP43 antibody to kind of be able to segment out which part of the tissue has lost the TDP43 function. And then we work with the core to run a whole human transcriptomic. I think around 17,000 genes were captured, and this is an image with basically every tiny, it's really hard to see, but every tiny dot on here is one transcript, and the color indicates the automatic clustering from the 10x procedure, and it does align with the tissue morphology a little bit. So later on, we did a differential gene analysis between the subpopulations of neurons with and without TDP loss of function. And basically, this is a graph showing the log2 ratio of TDP43 negative neurons compared to TDP43 positive neurons. And these are some of the interest genes that are differentially expressed. And the red ones are the ones that correlated with the cryptic exons that we have found before. So this is on a transcript level that we see there are actually differential gene expressions among cell populations with and without TDP43 function. We also are piloting with Xenium in Situ cryptic exon probes. So here is an overview of the tissue. We also used human FFPE tissue. I believe this is a human hippocampus. And these are a zoomed in view of the tissue. And each tiny dot is one transcript here for the Xenium that we ran. And on the right here is the IF staining that we did on the tissue. Since it's non-destructive, we were able to overlay the transcript with the tissue. And so the first row will be the DAPI staining, and the second row is the TDP43 staining, and the third row is the neurofilament staining. So the yellow arrows are at the exact same locations across all three stainings. So, for example, this cell has the DAPI signal is different than the TDP43 signal, meaning that there's a nuclear clearance of TDP43 function. So normally when TDP43 functions correctly, it's within the nucleus. And when it starts to lose its function, we start to see nuclear clearance and possible cytoplasmic aggregation in diseases like ALS and FTD. So the little dots here are example transcripts from one gene that we had on the Xenium add-on panel for this pilot run. And the red dots are correlating transcripts, and these are actually also cryptic exon transcripts. So the red dots are correlating with cells that has lost its nuclear TDP function, and the blue dots are the one that does not correlate but belong to the same cryptic exon gene. So this is just to show that there are some cryptic exon transcripts that actually correlate with TDP43 loss of function. And also feel free to interrupt me if there's any question. So this is another way to show that basically, for example, this gene has also a cryptic alternative poly A. And this is to show like how we can potentially design Xenium genes, because Xenium is fully customizable to detect the expression of the cryptic exon. So basically, in this diagram, the gene is being transcribed from right to left, and the black signal is a wild type cells, and in the red signal is in the cells with knockdown of TDP-43 and inhibition of NMD. So this peak here, the signal, indicates a cryptic alternative poly A. So in this case, if we have three different probe sets, probe set 1 is the cryptic exon that is actually only expressed when TDP-43 loses its function. And the probe set number 2 is the 3' mRNA, which would be expressed when there is normal TDP function and would have much less expression when TDP-43 loses its function. And in comparison to 5' mRNA in this region, this is the normal mRNA region, so it should be not affected by TDP-43 function. So this is just an example to show that by designing different probe set against different regions of the transcript, we can see different expected signal change when there's disease pathology. And we also have run a pilot Xenium run. So basically, the XY are just the location of the transcript. And on the left is the APP signal, which is basically also dense in neurons, but also in glia. It serves as a general number of transcript marker. As you can see, in these regions with the curvy lines, there's a lot of transcripts that are detected by the Xenium. And on the right, the color is a bit faint. So it's hard to zoom in, but on the right here, each tiny faint dot, the color dot on the back, indicates one of the cryptic exon transcript add-on that we detected. And the black circles are actually where more than 4 cryptic exon transcripts correlate. So we're thinking that it could indicate that these are the neurons where there's a lot of cryptic exon expression. And that would help us with downstream analysis of pick and choose which cryptic exons we would include in the downstream analysis. And one important takeaway between these two figures is that the black dots actually do not correlate strongly with the high transcript expression, meaning that it's likely not artifact of how many transcripts are in these areas. So currently we are doing a full custom panel of 300 genes and all cryptic exons, and to see basically which cryptic exons are the most indicative of TDP-43 loss of function to help us with our downstream analysis. Yeah, that was all I had. Thank you, guys.
Do you have any question for Angela? Sounds good. It's going to be around anyway, and it's wait, question.
Yes.
Thank you very much. The cryptic exon was this an isoform?
Sorry?
How did you actually measure the cryptic exon specifically? Was it an isoform we're looking at?
Yeah, by isoform. So by isoform, do you mean like the different part compared to the normal transcript?
Yeah, how did you actually design the probe?
Oh, okay. Yeah, so let me see. So I think, for example, let's see. Okay. So for example, this is the signal that is on the cryptic exon. So there's a lot of ways you can design it. So in this case, you can directly design against this region. So basically, this probe set 1 should only express when there is cryptic exon expression. There's also a lot of other ways. I think there's another way where you can say design a cryptic exon that should bind to both the immediate upstream and downstream of the cryptic exon. And in that case, only when a cryptic exon is not expressed would you see the panel signal, if that makes sense. So there's a lot of different ways. Yeah, thank you.
Okay. Well, thank
Thank
you,
you.
Angela, very much. Angela summarized basically all the things that Brad has discussed in one single experiment. We're going to have also later another speaker who's going to touch upon Xenium applications. In the meantime, I think it's lunch break, and it's a little early, but still you can enjoy it. And we'll see each other here at 12 o'clock for next presentation. Okay. Thank you very much again.
Yeah, for first feeding. Okay. But I mean, for some cells, like muscle cells, the nuclei is not going to defend me. So in that case, let's say it's slow, go through cell. Yeah, so I think muscle is one of the harder cell types. I think if we're doing the Xenium segmentation, I don't know, would any of them membrane stains do there? I think they do stains in those muscle cells. So I think that would be hopefully something we could kind of involve more. And then it's very clear. And there's regions, like you said, where the nuclei is and it's not that specific kind of signalized area. So muscle cell is important.
We have here John Lee that is going to discuss some of the issues that we have with issues. I guess what's the best practices for special assays tissue prep, which it is a very important part of the workflow. So thank you, John, for spearheading this talk today here. Thank you.
Thank you. So just to introduce myself, for those of you who haven't met me in person before, my name's John Lee. I'm the spatial field application scientist from 10X, and I cover Virginia, Maryland, and a few West Virginia accounts. I've been working with the core here for quite a long time. And I wanted to include this part of the talk into this seminar because, as mentioned, the sample prep, the tissue prep is really important for your assay to be successful. You have a core that has been working with 10X products for a while now. They obviously know what they're doing at the bench. And you will most likely have a bioinformatician in your lab, or you're working with a bioinformatics group that will provide the data analysis part of the assay. So all of that back end is covered, I believe. Now, the tissue preparation, from what I understand, is a lot of the cores, not only at Johns Hopkins, but in University of Maryland and other accounts. Tissue preparation is usually done by individual labs. You will section, you will do the staining, the required stainings, and you'll provide the sectioned tissues that have been stained and imaged to the core, and they will work with the tissues that are already on the slide. So it's really important that you prepare your tissues correctly. So the old saying, garbage in, garbage out. You want to make sure the tissues that you're committing for this assay is of high quality. And in order for the tissue to be high quality, you have to prepare it correctly following our protocol. So I will talk about the Visium HD and Visium HD 3' tissue preparation first. And then we'll move on to the Xenium tissue preparation. But most of the stuff that I'm going to talk about for FFP sections and fresh frozen sections, that relates to both Visium HD and also for Xenium. So just keep that in mind while during the presentation. So for Visium HD, as you can see here, there's a detailed overview of the workflow, but I'm going to focus on the very first column here where it says sample prep. Both FFPE and fresh frozen samples are compatible, and also fixed frozen is compatible for Visium HD. Before we go ahead with the sample preparation presentation, I want to show you a quick resource that we have available for you. This is called a quick reference card. Every CytAssist instrument is supplied with one of these quick reference cards. So if you are an instrument owner, you will have this laminated paper that was supplied with the instrument. If you no longer have this, or if you're not an instrument owner, you are able to download this quick reference card from our website. So please go ahead and locate this, because this quick reference card is very helpful for you to get started with sectioning. This quick reference card has a 2-scale template, which basically guides you to where to place your section on your regular glass slide. And as you can see in the animation to the right, once your section is placed on your regular glass slide, you can place your slide with the tissue section over this template to make sure that tissue is within the green allowable area, because that means that that slide with that section is compatible with the tissue slide stage on the cytosist. So the diagram on the right side is showing the schematic view of the cytosist tissue slide stage. So that's where your tissue slide is going to be aligned on the instrument. Another tool that we have is called a cytosist alignment aid. This is not included as part of the kit that you would purchase for the assay, or is not part of the instrument purchase, but this is something that you can purchase after the fact. But this acts as a guide to mark the tissue slide with the markings that you see on the right, so that those markings can be utilized to align the tissue slide onto the tissue slide stage on the cytosist. So now we will talk about the FFPE tissue preparation for Visium HD and also for Xenium. As I said, both HD and Xenium are compatible, or FFPE tissues are compatible for HD and Xenium. So we have tested a wide range of tissues for both human and mouse. We have a comprehensive list of the tested tissues on our website. So please make sure that before you start the assay, go on our website and check if your tissue is listed as one of the tested ones. And if the tested tissue, or if the tissue that you're interested in is not on the tested list, it may be in the challenging tissue types that we have tested. It does have note section on it, and it describes why it's considered a challenging tissue. So please do take a look at this before you commit your samples for the assay. So for the FFP workflow, there are 2 types of inputs that are compatible. One type is the archived FFPE H&E stain slides, and the other is the FFPE tissue blocks. Whether you're working with the archived H&E stain slides or FFP block, you will need to perform RNA QCing by performing the bulk RNA extraction and looking at the DV200 percentage. So as you can see, for both types of tissue inputs, you are converging into performing the tissue QC. And the QC metric that we're looking at is, as I mentioned, is DV200. And the DV200 greater than 30% is compatible and most likely would generate high quality data. When you're working with H&E stained archive slides, you will first image the tissue. You will need to generate a TIFF or JPEG image of the tissue with at least 20x objective. Then the hard set cover slip will be removed following our protocol. We have a protocol built into our tissue prep handbook. We're basically using xylene to remove that hard set cover slip. Then you will perform the QC. The tissue will have to be scraped off entirely or partially. So if you have serial sections that are H&E stained and archived, you will dedicate one of those slides to do the RNA QC. So you will remove the hard cover slip, you will scrape that stained tissue off that slide, and you will do a bulk RNA extraction, and you will take a look at the DV200 on that. If you don't have a serial section, you will have to remove the cover slip, and you will have to scrape off a partial tissue from that slide. And you will use that small tissue for RNA QCing, and then dedicate the rest of the tissue that's still on the slide for the assay. Once the RNA QC is done, then you will proceed to destaining and decross-linking steps for the slide to begin the workflow of Visium HD. Similarly, for FFP tissues, can be the FFP blocks can be sectioned into scrolls and collected for RNA quality check. Again, you're looking at the DV200, and once the tissue passes the QC, the tissue block can be sectioned, and that section can be dedicated for the workflow. For FFPE, we generally recommend sectioning at 5 micron thickness, but we have tested between 3 micron all the way up to 10 microns. Now, how do you decide what section thickness you want to go with? I always recommend sticking to the 5 microns, but if your cell is large that you have to section thicker than 5 microns, then you can section up to 10 microns. But the downside of sectioning thicker sections is that there's a higher risk of detachment. As you go up the thickness, there's a higher chance that your section might come off the slide during the workflow, during the H&E steps, during the probe hybridization overnight, during the entire workflow. There's a higher chance of your tissue falling off. So it's not always best to have more material on your tissue. So just keep that in mind. Now let's talk about fresh frozen tissue preparation. And for this section, I want to mention that fresh frozen is compatible for HD, compatible with Xenium, and also compatible with HD3'. HD3' is our non-probe based approach, and that assay is only compatible with fresh frozen. So again, we have tested a wide range of tissue types, both human and mouse for HD, and not only human and mouse for HD3', but for other species, because it's not probe-based. We're not restricted to the species human and mouse because this is not probe-based. You can throw on any type of species you can imagine, and yeah. Again, there are 2 ways to prepare the inputs for fresh frozen. One is freezing the tissue and then embedding that frozen tissue in OCT separately. Or you can freeze and embed in OCT simultaneously. And I will go over the 2 methods on the next slide, but here I want to note the stopping points that you see on the diagram here. Once the tissue is frozen, that frozen tissue block can be stored in -80 long term. We don't specify how long it can be stored, but it's for long term once it's frozen. Then that frozen tissue can be embedded in OCT, and once it's embedded, that embedded tissue can be stored in -80 long term again. When you when you freeze and embed together simultaneously, again, same thing. That can be stored in long term in -80. Once these are sectioned onto a regular glass slide in a cryotome, that slide can be stored in -80 up to 4 weeks. So those are the stopping points for the fresh frozen. And these are good stopping points, a good handoff point where you can hand off the slides to the next group that will be in charge of doing the following work. Okay. And again, and going back to the 2 different methods, this diagram is showing a detailed view of what's happening here. So the first one, simultaneously freezing and embedding. So basically, you're going to embed your freshly collected tissue in pre-chilled OCT in a mold, in a cryo mold. and you're basically submerging that cryomold with your tissue, with your OCT surrounding the tissue in a pre-chilled isopentane. Avoid freezing your tissue in liquid nitrogen, because liquid nitrogen will damage your tissue. It will cause a lot of bubbling, and it will cause a lot of crystallization of the moisture in your tissue. So always use isopentane that's pre-chilled. You can use dry ice, pre-chilled isopentane, or you could use a bath of liquid nitrogen to chill the isopentane. Then the second part, or second method, where you can freeze the tissue and embed separately. You're taking the freshly collected tissue, you're freezing it with isopentane, and then embedding that with OCT in a cryomold separately. The reason or the justification for picking either one is, whether you have a frozen tissue already in your disposal. So if you have these frozen tissues in your -80, then obviously you would have to go through the second method where you're doing the separate freezing and then separate embedding. But if you have access to fresh animal and you can collect freshly, I would always stick to doing the simultaneously freezing and embedding, because that limits the handling of the tissue. You're handling it much less when you're doing this all together. Once the tissue is frozen and embedded, you will obviously go into the cryotome to section. Always chill your block, pre-chill the glass slides that you're sectioning onto, pre-chill the tools. You'll be using the brush to, pick up the slide or pick up the tissue. You'll be using the brush to, flatten out the sections. Always pre-chill all of that before you start sectioning. The sectioning thickness, the recommended sectioning thickness for fresh frozen is between 10 to 20 microns. We recommend 10 microns because frozen sections are much harder to work with than FFPE. So it's thicker than the 5 micron that we recommend for FFPE. So here you can see the technician here using the brush to flatten out the section. You can use the anti-roll plate if you want. The only thing I ask is that before you start sectioning, it's a great idea to RNA zap everything in there, clean it up. Even the brush, I've had a couple customers who are washing their brushes, wiping their brushes with RNA zap. Wiping the stage and the anti-roll plates with RNA zap. So just keep in mind that you're working with RNA, especially when you are submitting these blocks to be sectioned to a histopathology department. They're not used to these RNA work, so always just remind them that the downstream workflow is RNA. You will pick up the section using the glass slide. You can aid the tissue section pickup by warming up the slide with your thumb. Some customers like to have the slide outside of the cryotome because that keeps the slide warm and it helps to pick up the slide section inside the cryostat. I don't recommend doing that. Again, because we're working with RNA, I want everything to be chill. I want everything to be RNase free before the tissue touches the slide. So pre-chill the slide in there in the cryotome. Just warm up the side or warm up the region where the tissue is going to be placed using your finger. Once the sectioning is done, protect your block, the remaining block, by putting more OCT on top of where the section was done. And this block obviously can go back into your -80 for long-term storage. A little bit more about the RNA quality assessment. You will section a few scrolls, section enough that will give you 20 to 30 micrograms of RNA. You will do bulk RNA extraction, and you will check the RIN score. So unlike the FFPE, for fresh frozen, we're looking at the RIN score. For HD, the RIN greater than 4 is recommended. For HD3 prime, which is a lot higher sensitivity assay, we recommend using tissues that give you RIN score higher than 7. If you have sections that are lower than 4 and lower than 7, I mean, there's nothing stopping you from using those samples. But what we have seen is the samples that have higher RIN tend to generate better data. But we also understand that, especially if these are clinical samples, they're very precious, and those are the only samples you have to work with. We have a few other optional tissue QCing methods. We recommend doing a DAPI stain on your tissue just to see how your nuclei look. So the thing that we're looking for here is looking for the nuclei that are intact. Your DAPI, your blue is, whole. There's no empty spaces inside the nuclei. There's no empty blue spots or empty spots in your blue stain. Tissue sections that have really strong DAPI stains and really filled DAPI or filled nuclei with DAPI stain tend to generate better data. We also recommend doing an optional H&E staining. Once you do the H&E staining and you image, you're selecting a tissue block that does not have, these cracks or freezing artifacts. And you're also avoiding the area that has hemorrhage, a lot of blood cells, or necrosis. This slide I just included to show you that colder doesn't always mean good. When you're sectioning in the cryotome, the sectioning temperature is usually around -20. But if you go colder, it might cause tissue tearing. So if you're sectioning and you're noticing that your tissue is tearing, there's a chance that it might be too cold. It's a good practice to try playing around with the sectioning temperature as well. Okay, and now we're going to talk about fixed frozen tissue preparation. And this fixed frozen tissue type is compatible with Visium HD. Unfortunately, Xenium is not compatible with fixed frozen, and neither is HD3 prime. The only tissue type that is compatible with 3 prime is obviously just fresh frozen. But fixed frozen is compatible with Visium HD. And again, we have tested a lot of tissue types. And we have the list of the tested tissue types on our website. So please go take a look at that before you start. And this is just a general overview of what's going to be happening for your fixed frozen tissue prep. So you're basically going to be fixing your tissue using a fixative. It's going to be a 4% formaldehyde or 4% PFA. And once the tissue is fixed, you're going to cryoprotect it using the sucrose gradient. And once the tissue is protected with sucrose bath, then you're going to embed it. And the sectioning is basically the same as fresh frozen sectioning. So here is more a detailed view of what's going on. So you're going to be fixing it with 4% PFA or formaldehyde, and you're going to put it in a 30% sucrose bath to cryoprotect it. And you're going to embed it with OCT, and you will section, and that section can be stored in -20 for up to 2 months. The OCT embedded block, again, can be stored in -80 for long term. And so fixing generally takes anywhere between 12 hours to 16 hours in 4C. A lot of customers ask me, how long should I fix my samples? I can't really help you there. You are the expert on your own tissues. So try to optimize what duration of fixation is best for your tissue, and always check by doing stainings, looking at the morphology of the tissue. But generally, it's overnight fixation in 4 degree. And you're going to fix in a tube with shaking, and every now and then just make sure that the tissue is sinking all the way to the bottom, because that is going to tell you that the tissue is properly fixed throughout. Once the tissue is properly fixed, you're going to take the tissue out, and you will do a cryoprotection in 30% sucrose. Again, this will take anywhere between 6 to 12 hours. And once the tissue is properly protected with sucrose, it will again sink to the bottom of that 30% sucrose. So just keep that in mind when you're wondering if it's been done. And once the tissue is properly fixed and cryoprotected, it can be frozen using the same method as the fresh frozen samples, embedded in the OCT with using the cryomold. The RNA QC is also performed on fixed frozen samples. Again, sectioned enough for 20 to 30 micrograms of RNA, perform the bulk RNA extraction, and look for the DV200 percentage. Because this is fixed, again, going back to the FFP, we're looking at the DV200 score. DV200 higher than 50% is recommended here. Now moving on to the Xenium workflow. So as I mentioned, Xenium is compatible with fresh frozen and FFPE sections. Our newest product, Xenium Protein, where you're looking at the gene expression plus protein using Xenium, that is currently only available or compatible with FFPE sections. But I believe our R&D team is working hard right now to validate fresh frozen workflow as well. So tissue preparation for V1 and Prime. Prime is the 5,000 panel. V1 is the 300 to 400 panel. Those two assays, the fresh frozen and FFPE tissue preparation, is very much similar or very much the same as Visium HD. So I'm kind of skipping a lot of those slides because it's exactly the same as the Visium HD. But the only difference is, instead of sectioning your tissue on the regular glass slides, you are going to be sectioning those sections onto a Xenium slide for Xenium. So here I have a schematic of the Xenium slide with the capture area in the middle and the fiducial marking here, you see. It might be really hard to see this when I go to the next slide, where there's a picture of a real Xenium slide, but just know that there are fiducial markings around inside the capture area. And these fiducial markings are used to align multiple images that the Xenium instrument will be taking during the run. So it's really important that you don't cover those markings with your tissue when you're sectioning. Similar to Visium HD, we have a 2-scale template of the Xenium slide with the area where your tissue should be placed on. So what you can do for this is you can take a regular glass slide, put it on top of this 2-scale template, trace out the capture area, and just practice landing your tissue in this capture area before you proceed to sectioning onto the Xenium slide. So here is a picture of Xenium slides. You can see the fiducial markings inside the imageable area. You can see those white dots there. So those are the dots that you want to avoid covering. If you do end up covering it, it's not the end of the world. We do have a protocol where we guide you to scrape that tissue off to reveal those markings. So it's not the end of the world, but the more you handle this tissue slide or Xenium slide, the higher chance that your tissue might come off during the instrument run. So just, good practice to section and avoid those areas. And obviously, you can section more than one tissue on there. A lot of customers ask me, what is this gap between the two lines here? I don't know why there's that gap there. It's just there. But you can section your tissue across that gap without a problem. Now, going back to the schematic, I'm kind of going over this in Access, but again, try not to cover those fiducials. The fiducials at the top and the bottom are especially important. If you cover those, you almost certainly have to scrape that off before you load your Xenium slide onto the Xenium instrument. If you cover a few fiducials on the sides, it's okay. The instrument will have enough fiducials to align multiple images. Here we have good and bad examples of tissue placement on the Xenium slide. So good examples are those that are not covering the fiducials. You see this tissue section with excess OCT maybe covering some of the fiducials here. That's okay because we have a deparaffинization step in our protocol that will remove that excess paraffin. So just make sure your tissue's not covering those fiducials. Going to the right here, looking at the bad examples, you have tissue obviously outside of the capture area and also covering the fiducials. If you run this, it's not going to error out. It will run without a problem. But the problem is you're going to be missing the data coming from the tissue outside of the capture area. So you will generate data from here, but you're missing almost half of your tissue. Here, same thing. There's two tissues over top of each other. It will generate data. The instrument will run. But whatever transcripts you're seeing in the middle, you don't know where that transcript's coming from, whether it's coming from this tissue or that tissue. This I think would be an issue because there's tissue covering the bottom fiducials here and tissue covering some of the fiducials at the top here. And again, there's multiple tissues on top of each other. So. There are other QCing steps for Xenium. Again, similar to the H.D. workflow, we recommend doing a DAPI stain to make sure your nuclei is whole and DAPI stain is strong. You can also do an H&E staining on it to see there's no artifacts and there's no cracks or folds on the tissue. You may also perform a DV200 to see how your RNA is, how fragmented it is. DV200 score less than 30% is correlated with low quality data. For fresh frozen tissues, again, DAPI stain to look at your nuclei, do an H&E stain to make sure tissue morphology is there. We don't recommend doing DV200 here because, again, we're working with fresh frozen here. But for FFP samples, looking at DV200 is a good idea as well. So that is all I wanted to talk about today. So I've gone over tissue preparation for HD, HD3', and Xenium. So one thing I want you guys to leave with is, garbage in, garbage out. Make sure your tissue sample is of high quality because I deal with a lot of troubleshooting as well as doing on-site trainings. But a lot of troubleshooting ends up being, okay, your tissue quality was bad to begin with. So, okay. Thank you.
I can't stress enough what John just said. Please take care of the preparation of your samples. We have quite some experience in the lab with detachments from specific tissues. There is special slides that you can use, but somehow you guys refuse to use them. Not pointing fingers, but it happens. The DV200 more than 30%. I wouldn't like a 31%. We have results which are not necessarily reproducible when you are very close to the limit. You get results, just that it's not as good as you would if you get a DB200, which is more than 30%. It's just a matter of fact of, the sample being degraded. And the other thing is the RNA is free, indeed. Initially, when we worked with the core sections of the tissues, they weren't doing that, and that caused some troubles. So please stick to RNase-free processing when you need to do those. We use right now the OTIS core, which has a quite good collaboration with our core, and whatever they do with the samples, now it's what it needs to be done. But you can also do it yourself. Just be sure that you know what you're doing.
Hello, everybody. Thank you, Manu, for giving this opportunity to talk about the work we do in the lab. And today I'm going to talk about Xenium in situ. And we're going to talk about different aspects of spatial transcriptomic using this technology, particularly in the context of natural aging in the retina. I'm going to present some also challenges to study high, like super dense or compact tissue, just to resolve different gene expression profiles at the cellular and space level. And finally, ending with some conclusion remarks. So we studied the retina, and we know that the retina is basically the door for reality for everybody, right? And the retina, it's this, for people that's not in the retina field. Let me see if I can move. Okay, it's here. It's this tiny or thin layer that's sitting behind the eye. And that let us explore different things, right? And it's a highly laminated, organized, beautiful looking tissue, as this is drawn in Ramonica Hall pictures from many years ago. However, we face different challenges when we observe this tissue at the cellular level using microscopy. And particularly it gets more challenging to assign different transcripts into their different specific cells. In this context, today I'm going to talk about this study that it's a preprint, and it used some comparative single-cell multiomics analysis to see if there are different evolutionary conserve or species-specific cellular mechanisms that might be mediating aging. And this is a study led by a former postdoc on our lab, Isabella, together with Ping. In this study, our group presented an atlas through different time points in 2 different animal models, in zebrafish and mice, over all these different points. And we also use human aging results already available. For this talk, I'm gonna show some spatial transcriptomics data for this particular points in mice, since Xenium is only available right now for mice or human. All right, so what we found basically many different genes that are changing, of course, right? But many of them has suspecting different profiles, some of them decreasing while aging, some of them increasing, and some of them going up and down through the course of lifespan. Together with that, we found that the probably major pathway or conserve way of that people, it's basically aging. It's like the increase of inflammatory markers and autophagy markers through the whole different tissues, particularly in the in Mueller glia, which is a supporting cells that promotes healthiness, we would say, into the different neurons in the retina, including photoreceptors, bipolar, amacrine cells. So despite of analyzing and including different gene regulation happening at different cell type, we also were able to develop some aging clocks that can let us estimate different biological or molecular age in different samples. They presented different aging clocks for each different cell types through all those 3 different models included, using linear regression models. Together, this is actually important because we can track maybe later when we are trying to do some therapies to reverse aging, or to promote, or to look for some therapies that look for rejuvenation or even stop it. Particularly, we saw that there are some changes happening at the molecular spectrum, but are not only related to gene expression profiles, but also by the different area localizing, particularly in zebrafish. This means like, well, there are different areas that may be aged differently in different time points. So we wanted to see whether these changes that happen in zebrafish also happen in mouse. I'm going to talk now about some results using Xenium. Probably you heard about this technology a lot today. It relies on probe hybridization, and if they align correctly, we will be able to, after ligation, amplify this signal that then later it's decoded by a set of like hybridization with fluorescent probes. And with this decoding, we have the ability of assigning each different transcript to a specific point in the tissue. So, okay, we got the different points in the tissue, but like we cannot differentiate the different cells that are located currently on the cell. So this is something that was challenging, particularly for dense tissues, but we work on a pipeline to improve this kind of imaging quality using another type of microscopy that it's now available using the module from Apple from microscopic size, that we will able to get some better, not better looking, but like at least individually separated cells that we then could be able to assign manually. We face different time just to perform manual alignment of and registration because these are images taken from 2 different microscopes. We spend a lot of time doing that, and to then later align and perform an actual image that can reflect the right cell localizations. This is the results. Well, Xenium has really nice results in many different tissues, but in the retina it's kind of challenging right now. We can see that basically the inner nuclear layer, it has only the tissue that gets segmented, but the whole area with photoreceptors, you don't kind of see or differentiate any cells. By using this pipeline, we were able to resolve the different points on different cell types and be able to assign those transcripts into them. With that data, we now were able to see and separate most of the major cell time points in this Xenium tissue, including photoreceptors that are normally photoreceptors and cone that are in the outer nuclear layer, and retinal ganglion cell here in yellow, as you can see here, and the other cell types in the inner nuclear layer, including amacrine, bipolars, etc. So with this data, we also perform aging clocks, because this will, as I said, this will set the ground to see how age progresses over time as a molecular unique profile that we can later infer into different samples. Just to show the comparison between single nuke RNA-seq data and Xenium data on Muller glia, you can see even though we use only 4 time points, we were able to basically see the same trend happening at the single cell level with RNA-seq. And if you can see here, these are the profiles for actually those genes that gets increased over time. This is in mouse. Together with this data, in collaboration with some analysis, some group that performed like analysis, they were able to make this kind of like really nice setup to test some cell proximity effects. And we need to, the single cell data from single nuke RNA-seq can give us a really nice profile or gene regulatory network analysis that we can infer something, but we are losing the spatial interaction that might be happening between cell and cell. So with this analysis, what it does is trying to see what happens if we choose one target cell, what's happening with cells surrounding them, and maybe if their expression profiles changing in other farther away cells. So we can choose a target cells and effector cells around them. Based on that, now that we have a predicted aging clock, we can assign some aging velocity to each cell by just comparing the molecular age that it's predicted with the average age that's supposed to be happening in the whole tissue. So given that age acceleration, we were able to find that particularly cells that are, let's say, rod on bipolar cell that are next to cones or rods tend to have a more pro-aging profile. So in the sense that cone or rods might be exerting these kind of aging properties, right? But interestingly, when rods bipolar cells of amicrescence are surrounding Muller glia, they tend to have a more pro-rejuvenating effect. I don't know if you remember what I said at the beginning when we checked for the molecular profiles in single cell. It actually shows that there were like a lot of like inflammatory pathways. However, in Muller glia, however, here we're seeing that being close to a Muller glia is actually good because it might be reversing at least molecular age. So these data tell us that cells maybe are interconnected and that in aging rates might be influenced by nearby cells. We also explore some different region-specific expression changes by classifying different regions in the retina between the periphery and the central. So this is given by, because in zebrafish, it was seen that those two different parts behave different during aging. Particularly, Muller glia or glial cells were increasing their glial quiescence markers at the end of the transcripts of aging. So as you can see here, if we look in this axis, it shows more of the central retina, and here what it's closer to the periphery. And here you can see an example that for bipolar cells, these markers, it's more enriched in the central. However, for example, here in Muller glia, this marker related with inflammation, CD36, it's more enriched into the periphery, something that you can actually see through the progression of the disease. So it was pointed. But okay, here you can see bipolar cells. Here you can see the central retina and this marker Greek 3. And for example, Muller glia CD36, it's more prominent or more correlated towards the peripheral. And you can see how this gets spread through lifespan on mouse. So given this, we now are showing that there are some regional differences through the aging course. And yeah, that's kind of something nice. But like, I don't want you to live with the idea that, oh yeah, this is like, I don't want to be manually annotating different cells to be able to resolve these spatially distributed genes. So Xenium came out with this actually nice technology that has some antibody staining this extra step to do some staining to see different boundaries of different cell types to stain for different markers like boundary protein staining, interior RNA staining, and some interior protein staining. So we can see each of those labeled and combined together. And this is what you can see at the Xenium website and how the cells will look at that. But again, when we look into the retina, still, it's challenging because of how dense the cells are. And this is not only a problem with this platform, it's a problem in general in spatial transcriptomics when you try to segment and assign different cells to cells that are really actually super compact. But thankfully, for our luck, a new article came out with new models now it's the boom of AI. And now we were able to use some different segmentation pipelines that were available to actually use this multimodal image that we have from Xenium to retrain our own retina tissue and see if we were able to separate different cells and assign them different transcripts correctly. And it actually performed really well. So now we don't have the need to do the re-image or alignment for different tissues. You can see here what was something that was obtained by the raw data with Xenium. And here is something that how we can see after the training model when we optimize different settings. So with that, we now can use this powerful technique to now profile new samples that we have in related with other pathological events happening in the retina. So I want to conclude saying that spatial transcriptomics reveal these region-specific agent signatures and different cell proximity effects, Mueller-glia exact pro-rejuvenating influence in nearby neurons, and there are available different there's like a different tools that we can put together with the Xenium staining to actually get new data and improve cell segmentation. I want to thank our lab and our collaborators, and thank you for your attention.
Thank you, Roger. Is there any question for Roger? That was hey,
cool.
One is there.
Hi.
What's the using all the mineralities to mention and those from Xenium, or was it just the
So, sorry, I didn't hear you well. And I know you talk about different segmentation methods.
yes, the segmentation that worked best for you, the retrain one. Was it based on the image or
On this? No, it was based on the new image that we took using a different scope. So we image on a size apotome microscope that has like a system that basically do a better deconvolution for different spots. And we use an already trained model available in QPath that it detects basically circles on the model. And that's what we use to segment the data that I just presented. The new available model, it's like from CellPod that they just upgraded. And that's from all other data that we're currently analyzing. Yeah. Sure. All right, cool.
Thank you. Thank you, Roger. That was great talk. Not only thing retraining of the model AI brought to your results. It was really impressive. And now we have Brad Thomas again. Brad, it's going to give us some information about demonstration of the data analysis.
Well, thank you everyone for sticking around after lunch. The goal of this is to kind of go through now a software demo. So I have about 15 minutes. Please bear with me. This is an online demo, so there's a lot of data, a lot of information. Sometimes the images do take a little while to load. But before we kind of get into kind of the demo here, I wanted to pull up the 10x webpage and show you where I am getting this data from. So I think kind of one of the benefits of what we have here at the 10x website is all the information typically is publicly available. All our software tools are free. We have online data sets. So you can see here I have the 10x Genomics webpage pulled up. If you click on the support tab, you can scroll all the way down to the bottom. You can actually up top, this is where we're going to find a lot of our kind of wet lab support, kind of protocols, best practices, those type of things. But for the purpose of today, I'm going to scroll down to the bottom and you can see we have a lot of our software tools here as well. So clicking on Xenium Explorer, what we can see here, you have your free download where we can download these tools. And then I think something that's really powerful as well is we have a tutorial section here too. So the 10x team is always happy to kind of meet with you to show you how you can use these tools. But I think this is always a good spot for kind of self-learning. Download the tutorials, download data sets, and you can start exploring. As far as the data sets we're going to work with today, you click on the Xenium Explorer demos, and we have a wide range of different demos here as well. And if we actually go to the regular support page and data sets, you can see we have 44 different data sets that you can download and start playing around with yourself. So with that being said, we're going to start with this ovarian cancer data set. Again, the images do take a while to load, so I've zoomed in first and then we'll kind of back it out. But we have talked a lot today about kind of the cell segmentation software. We heard Roger kind of talk about kind of what some of these components are. As Roger said, the benefit of this is this is going to work with a lot of tissues, but it's not going to work with everything. And for those tissues it doesn't work with, you do have the ability to create your own segmentation masks and import them into our software. But currently, in looking at this, you can see here we have a variety of different drop-down menus. I have the images tab pulled up, and we're looking at Xenium morphology, where we can start to see some of those markers and start to see these true segmentation aspects here. If we looked at them one by one, we can start with looking at the nuclei. So this is your DAPI-based stain. We can go through and start to look at the boundary stain. So as we talked about earlier, this is a hierarchical approach. We want to kind of use this first to start to understand kind of where those boundaries are at. If that doesn't work, we go to the interior stain, right? This is going to stain now our 18S, where we can start to think that's going to fill the cytoplasm. And now you can kind of start to infer where the nuclei are at, where some of those membranes are at as well. And then at that point, if neither of those methods work, then we go to the nuclear-based stain and we can start to go through and do an expansion on that. But kind of looking at it all together, we can start to get this compiled image, and I think kind of a very nice representation of what that biology looks like. So now I'll zoom back out a little bit so we can kind of get a bigger full picture of what this looks like. So again, this is an ovarian cancer. You can see here a lot of the tumor here. This is where a lot of that segmentation's at. You can see down here in some of the like the stromal and kind of structural regions, we're still getting a lot of cells here, but again, the view is going to be a little bit different than we're talking about now, where the cells are at here in morphology-wise versus kind of these densely packed regions. As far as other aspects of this tissue, we see the ovaries up here as well as the fallopian tubes. So for me, I think in kind of looking at these data sets, the first thing I always go to the web summary here. We can click on this information button. It'll go through and kind of give us a breakdown a little bit about the sample, what panels were used. And if we click here, this is going to be the web summary which we're used to seeing kind of with our single cell and Visium aspects, where we can start to understand the number of cells. Median transcripts per cell is here. I think this is a little bit misleading at times because, again, this is going to depend on the biology. We think about those regions in the tumor. I'll show some of this in a minute, but typically a lot of those cells are getting to 500,000 plus transcripts per cell. We start looking at some of the stromal and fibroblast regions, it's going to be a lot lower. So the average median transcripts per cell can sometimes be misleading, but it is a good kind of metric to kind of get started and look at. You can see here, the only one we're analyzing, this actually Xenium slide ran 2 tissues. We're only analyzing this portion up here. And then we can get some other high-level metrics here. Other tabs, if we go through and look at this, again, we talked about that optical coding aspect here of how we can determine kind of which of these probes are working and kind of what gene that is related to. And here it shows it great on a waterfall plot, right? We're looking at our Q scores here, as well on this axis here, starting to look at the total transcripts per gene. And we want the majority of now our gene probes to be up in this upper right-hand quadrant here. This means now they're high quality and we have a lot of them. If you see over on this side of the key, we have a lot of negative controls built in, genomic DNA controls. And again, we want those to see, you can see less than 10 copies here for most of those. And the ones that are there are very low quality, below Q20. Lots of other metrics here you can kind of go through and analyze as well. And then for the cell segmentation tab, this starts giving us a little bit of information about how well that cell segmentation mask worked. You can see here, about 28% of the cells were called down with the boundary stain, about 70% with that 18S stain, and then about 2% were performed with the nuclei expansion. And then the last tab I'm going to show here, again, some basic analysis here where we can start to go through and look at clustering and UMAPs and start to kind of get a high-level view of what kind of genes are present and what those log fold changes are within these different clusters. So I think that's always a good spot to start here with the web summary, but I know we kind of want to dive a little bit deeper into kind of what is going on in this tissue. Again, we've kind of shown the morphology here. We also have the ability to, again, through image registration, bring in the post-Xenium H&D image. This is a great tool now. I will show how this is working here in a minute for kind of understanding the transcripts that are there and how that matches up with the true morphology. And then from there, we also had an H&E image annotated by a pathologist, where we can kind of now import this in. The red regions are tumor. I believe the green are the ovaries. Blue is smooth muscle. And then this darker green here is going to be the fallopian tube. So we've had a pathologist go through and annotate this, and now we can start to incorporate that, as well as kind of these regions of necrosis, which will be important for some discussion here in a minute. Another thing, let's see if we can zoom in if this loads. I think it's a nice aspect here. Again, we can kind of see some of these segmentation markers as what they look like from the morphology view. But now we can start to turn on our cells and see kind of how well some of that matches up and make sure that we are getting some of so we'll turn the opacity down a little bit. You can kind of see those segmentation versus the cells. And if we find this nice blend, we can see that that segmentation mask really is doing a very good job of calling these cells here. Let's see if I can turn some of the images off now. If we want to go through, we have several different ways of clustering. I think the typical way, especially from single cell kind of approaches, is doing graph-based clustering, where you can start to kind of go through and use these algorithms to find kind of now those most similarly distinct transcriptional profiles and start to perform clustering off of that. We can go through k-means where we can pick and choose the number of clusters. In this case, looks like we've chosen k-means of 2, but we can kind of go through and edit that anywhere from 2 all the way up to 10 to perform any of those analyses. And then we can look at custom cell annotation. So here's the point where we've actually gone through, used annotation tools to go through and upload this, where now we can start to see specific cell types, right? We have these groups here. You can see ciliated epithelial cells, inflammatory tumor cells, and we can start to go through and use that transcriptomic profile to go through and perform the clustering of these particular samples. One region I really kind of want to focus on, if we zoom in here, this is actually these regions here in yellow, right? These are going to be these malignant cell lining cysts. So if we look at this, it's kind of a protective layer here, single cell layer thick right around some of the tumor cells, as well as some other cell types. But if we kind of scroll over to other regions of the tissue, we can see we have these particular cells here. These are going to be your SOX2 OT positive tumor cells. They're really not protected by that kind of lining layer here. We can also see, I believe these are VEGF tumor cells as well. So we can see a couple different tumor populations here within this tissue. And I think if we took this and lined it up with our pathologist annotated H&D image, let's going to turn it on. There we go. And we change some of this opacity, we can see that, these regions of tumor, when we bring that down, right, this is looking at that region of necrosis that the pathologist had already annotated. So I think this is a great way to go through and say, we're finding this biology from the pathology view. What does it look like from a molecular level, right? So this is kind of a confirmation aspect here of what we're starting to see. Now, the other side of that I kind of want to think about now is another region of the tissue. This tissue here, kind of these cells in white. So these are going to be inflammatory tumor cells. So we went through and looked at gene signature. I can show you what genes these are here in a minute. But this is kind of a region of tissue that now if we turn off that opacity, this is not really a region that the pathologist annotated. So I think this shows the discovery type approach of what we can do in the software, right? This is the last region we saw kind of those nuclei that look like they're going through and dying. Here we're not seeing that, right? This looks like normal tissue to the naked eye, to the pathologist's eye. But when we start to look at it from the molecular level, there's a lot more going on here than we can really determine from that pathologist view. So I think a great way to kind of think about how we can use these tools. From here, again, I've only shown the images and cells. Transcripts, this is a 5,000 gene panel, so we'll see if this works. It might be a little too much to load. Actually, right now I have it selected just for those tumor inflammatory cells, so 18 genes. Here's a list of what some of those genes are, CXCL10. I mean, this is actually one of those genes that was present in one of those publications I presented earlier, kind of thinking about some of those inflammatory responses. You can see the variety of different genes that we have clustered here. I'm really focusing in on kind of this region. If you kind of zoom out, you can see other small little pockets of this, but the majority, obviously, you can see is kind of highly clustered here. Now, another view we can do just with the 18 or just with the genes you have selected, which in this case would be 18, we can go up to now this per gene localization, and we get almost this view. Actually, I'll scroll to the bottom first. We get a view of all 18 of those genes, kind of in combination here, all the genes that are selected, and kind of see within the tissue where these pockets of gene expression are. But with this per gene localization, now we can kind of look at each of those 18 genes individually, see the number of transcripts that are present, and see where they're at within the tissue. And I think this kind of gives you now this holistic view throughout the tissue for very specific genes. So really a powerful view. in kind of where these genes are expressing. Other tools we can do, if again, kind of this is like, I'd like to think of this tool as a good secondary analysis where we can start to understand the biology, visualize where those transcripts are at. But if we want to take this and we find something interesting, such as this region we were looking at earlier, these inflammatory genes, we can start to go through now and start to export this data. So if we'll zoom in here, we can use our little lasso tool to create a freehand selection. And it might be a little messy here, but we'll see if we can kind of capture a lot of the gene expression we're looking for. And now this is going through and isolating this area where we can go through and we can start to export this. We can go through on a cell by cell level. It shows the area. It shows, again, what cluster they're in. And we can go to transcripts. We have transcripts turned on, yeah. Let's see, it's loading here to see if we can pull it up. But this should show us a list of all the transcripts that are present in that area we isolated here, if the software cooperates with us. It'll give us essentially to the point where we can export that into, similar to single cell where you get a cell by gene matrix. This is going to be more of a matrix now of location or kind of that cell ID by those genes that are present. And you can see here in this particular region, I have isolated just for those 18 genes. Again, this is not looking at the full 5,000. I think that would blow the software up right now online. We found about 96,000 of those genes present. And you can see the breakdown for each of those 18 genes. It looks like this MX1 gene is the most prevalent here, really about double what we're seeing any of the other genes here, with about 17,000 transcripts. And again, what we could do is we could go back and, well, on an individual cell basis, I don't have cells, points, cells. We could hover over specific cells and start to understand what is truly present at each of these cells as well. So I think I highlighted everything I wanted to show here. Is there anything I'm missing? So there's a lot you can do in these tools. So hopefully I haven't missed anything. Again, I know it's a lot to take in all at once. The 10x team is happy to kind of connect with you guys, go through these demos, start to make sure you can kind of understand your data. But I think the reality is, I think this is a great tool for better understanding the biology of your sample. And the way I like to use it is for the biologist to better communicate with their bioinformatician, so you can have those proper conversations. And then this information can then be taken into third-party tools, Seurat, STLearn, Giotto. There's a lot of other applications out there. But again, it's hard to have those conversations with the bioinformatician if you don't know enough about your own sample. And that's where I think these tools really give you this visualization and a better understanding of what that biology looks like. So hopefully that was helpful. It did seem to cooperate with us, so I've been happy to see that. But I think I'll pass it over. If there's not any questions, we'll take any while John's getting set up. I think John's going to do a software demo of some Visium data.
Thank you, Brad. That was great. I wanted to quickly demo the Visium HD data set here using our Loop browser software. So Loop browser, similarly to Xenium, is our software that's freely available on our website. This Loop browser is shared with single cell data, I believe. It's like a Xenium Explorer, but for Visium HD and HD3' data. And so here, the data set that we're going to be looking at is colorectal tumor sample here. So here we have a H&E stained sample, and you can see this little rectangle area here. That is where the Visium HD probes were hybridized onto and interrogated for the assay. So I'll zoom in, and it's very similar to what Brad had shown us with the Xenium data. You can see that there are segmented cells here within the data. So with our new Space Ranger 4.0 or higher, we have H&E stain-based segmentation pipeline built in. So it is able to segment the cells based on the H&E stain. It's using the pipeline similar to StarDist, so it's a nuclear stain expansion segmentation pipeline. And so you can see we have these segmented cells within the Visium HD data. And so similar to what Brad was showing, we can play around with the opacity here. So you can look at the cells, H&E image versus the segmentation of the Space Ranger pipeline. And I think it's doing a pretty good job segmenting the cells. And the colors that you're seeing is based on the clustering. So you can see here on the left, you have the clusters that have been generated by the Space Ranger. Default is the graph-based, but similar to what Brad showed, we can go into the k-means and kind of force the data set into a number of clusters. So we can click 5 here. We're looking at the data in terms of 5 different clusters. I like to work with k-means just because graph-based clustering tends to have a lot of clusters, which is kind of overwhelming, especially when I'm working with tumor samples. I like to have the clusters, anywhere between 5 to 10. That just makes it easier to take a look at the data. What I really wanted to show you here during this demonstration is going into the Features tab. And here you can search for certain genes that you're interested in, or you can go into gene expression sum. When you type that in, you're looking at the sum of all the genes that were part of your panel and looking at the expression level of that on your tissue. So you can change the scale here. Right now we're in log 2, but you can change it to linear scale or log norm. You can change the color scheme to whatever color you'd like. And so you can look at, which area of the tissue is transcript heavy by looking at the gene gene expression sum. Another thing I wanted to show you is you can upload your own list of genes that will help you annotate these cells. So basically, you can generate your own CSV file. So you can have an Excel file with 2 columns. The first column, you can name the genes. And the second column, just basically saying what those genes will annotate as. So here, I'm going to upload a file that I already have. And here is the file that I have. And basically what that file was an Excel file with the first column saying tumor or saying the gene names. And then the second column saying that gene is shown to be highly expressed in tumor cells, or this gene is highly expressed in goblet cells. This gene is highly expressed in fibroblasts. So I have this list built in an Excel file, and I can upload that. And so you can see tumor cells with the corresponding genes that I specified were highly expressed in tumor cells. You have goblet cells with the genes that are highly expressed in goblet cells, fibroblasts, and macrophages. So this is basically how you can annotate your data using the previous knowledge that you have. Hopefully from the 10x single cell assay you ran before HD gives you ability to annotate your cells here. So this, aspect of the loop browser is something that not many people have tried with their data. So I encourage you to try this out. Make your own CSV file and just upload it, name those columns correctly, and start annotating your data and just playing around with it. And one other thing I wanted to show you is you can go into the expression distribution. We have the tumor selected, looking at all the genes that are known to be expressed in the tumor cells. You can look at the violin plots, and that violin plot tells me that cluster 2 and cluster 3 are the clusters that have these genes highly expressed. So what that means is I can go back to the cluster tab, and I can start renaming some of these clusters as tumor. So cluster 2 looks to be highly expressed or highly expressing the genes that are specific for tumors. So I can start renaming this cluster by using this option here. And I can rename this tumor 1 and cluster 3 as tumor 2. So here you can, start personalizing your data by renaming based off of the annotation that you've uploaded. And I want to exit out of this. And similar to what Brad has shown us, we can select some of the regions in our data set. So this, I'm going to circle this area here. And it's going to allow me to rename that selection. I'm going to say selection. Finish. And so now only with this area selected, I can go into Reanalyze tab, which opens a new window. And basically what I'm trying to do is I'm going to try to recluster the region that I have selected. And so when I go through all these steps to fine tune my reclustering parameters, and making sure that a UMAP is there, and I'm going to name this region of interest and recluster. It's going to take some time to recluster just the area that I've just selected using the freehand selection tool, and it's going to give me a fresh set of clusters that are only within that region of interest. So instead of looking at the non-biased clustering that Space Ranger has defaulted to, or looking at the k-means, you can recluster based off of the region of interest that you have selected in the data. So let's wait for this and take a look at the clustering that this generates.
Hopefully those overlaps.
Yeah, hopefully, yeah, that's why I selected a smaller region. All right, done. And so you can see the UMAP view of the clustering that was redone, just selecting that area. You can look at, sorry, that was Tisney. Let's look at the spatial view and going into that area that we selected. And it's struggling. Yeah. So there's clustering within that region that we selected. So yeah, this is what I wanted to show you. Very similar to the Xenium Explorer. I love the fact that our new Space Ranger pipeline has the cell segmentation built in, which is great. The only thing you want to remember is to take advantage of the Space Ranger's segmentation pipeline for Visium HD, you have to perform H&E staining. This doesn't work for IF stainings. And also you have to have high quality H&E staining imaged. So I recommend doing at least 20x objective imaging if you want to take advantage of this. If your H&E staining tissue has bubbles or it's uneven, so it's blurry, the segmentation isn't great. So make sure you have high quality image if you want to take advantage of this. So yeah.
Yes, thank you.
Thank you.
That was great. I like the reclastering of the region of interest. That's really cool. Well, I guess this concludes the presentation part of the symposium. There's now, time for you to hang out with core, with 10x people, and ask them as many questions as you want. And we are here for that. So thank you again for participating to this. Thank you to all the speakers and their organizer, of course, Barb, Ajian, for being here. And we hope that this was informative for you. Thank you very much for being here.